clinical session this afternoon is Aaron Almeida, and we thank him for joining us from Arrowhead to talk about their program.
Thank you. So yeah, I'm pretty excited. Thank you again for the invite to talk here. So we're a little bit different compared to a lot of what people are doing here. So we're for dynamic polyconjugates is our platform for delivery of siRNA triggers. So our actual API is an RNA trigger, but we use a peptide as part of our delivery platform. So Arrowhead is in the field of RNAi and gene silencing. So this was discovered back in 1998, where Andy Fire and Craig Mello discovered that if you inject long double-stranded RNA into nematodes, the genes that have the matching sequence to that, you get potent silencing. And so this was an important discovery for just understanding how the biology works, and this led to a Nobel Prize in medicine. And so what people have worked out since is that these duplexed RNAs are chopped up and then they're loaded onto a protein complex that's referred to as RISC. When they're chopped, then you get this smaller fragment of it that's loaded onto RISC. It'll grab one of those strands really strongly, whereas the other one will be cleaved and discarded. And once you have that RISC protein complex with that single strand loaded onto it, what you have is a catalyst for the degradation of mRNAs, where the selectivity is defined by the nucleic acid base sequence. And so there's a lot of natural roles for the siRNA, viral defense being a big one. Traditionally, mammals have an immune response against these. But, and so the field of RNAi trigger is to feed in exogenous trigger so that it feeds into this pathway leading to mRNA knockdown of your target gene. But all of this needs to take place in the cytoplasm of cells, and that's probably a very familiar problem for this crowd. And so, as you might guess, delivery has been a key barrier for the use of the RNAi triggers in animals. So part of that is getting it to the right tissue, but then also from the tissue into the correct cell type in sufficient amounts. And so naked RNAi triggers are rapidly cleared by the kidney, and so you just don't get a large amount of RNA trigger there. And so people have worked out RNAi trigger small molecule conjugates, and I'm going to put cholesterol and kind of small sugars like NAG here along with peptide, just because it's a more analytically definable feature as opposed to some of the synthetic RNA trigger delivery vehicles like lipid formulations or some more synthetic polymer nanoparticles. So some of these synthetic RNA trigger vehicles are lipid formulations and polymers. Both of them can be really efficacious, but when we've gone kind of in the field of more polymeric nanoparticles because we feel that they're more just targetable, and that's where we've developed. So it's kind of a schematic of our DPC technology, and it's the role of it is actually to help the RNA get into the cytoplasm. No, I just said that, but and so what we're usually working on is an endosomal release pathway. It's a modular RNAi trigger delivery platform, and the kind of schematic that I'm showing here, it's actually a co-delivery. They're not interacting with each other. They're not complexing. They're just both injected at the same time. We use that for organs that just don't have a large PK requirement like the liver, whereas some of our other programs, like a renal cell carcinoma program, we can actually conjugate them together so that they arrive at the tissue together. And then we've also been working on an epithelial cell targeting program recently. As the DPCs themselves are targetable to specific cell types, and so you can end up with cell-specific uptake. And I'm going to kind of provide evidence for each one of these as we go. And then at this point, we have a clinically validated safety profile showing that it's non-toxic in humans. And so I kind of went pretty high level for a lot of this early on, some of our early proof of concept data, just to show the development of the DPC part. But because it's modular, where you can trade out your RNAi conjugate trigger, we weren't really working with a therapeutic trigger early on. We were working on kind of proof of concept things that are easy to measure. But then I'll go on into later a short story about our major clinical trial program. So if we're looking at our DPCs for liver delivery, our API will be an RNAi trigger that it has a cholesterol conjugate. These are canonical or other formats, a wide variety of RNAi trigger formats, always duplexed molecules, usually very highly modified so that there's not much ribose in them at all. There's a liver tropic cholesterol ligand just to get it to the correct tissue. And then co-administered with that is our dynamic polyconjugate, which we'll refer to here as NAG-MLP. And so it's an amphipathic peptide that facilitates endosomal release through membrane interactions. But kind of Arrowhead's niche has been making sure that it only interacts with the membranes under very specific circumstances in the endosome. And so we have the CDM-NAG masking agent to control that. The CDM masking prevents the nonspecific interactions of the peptide during systemic circulation. And then once you've blocked those nonspecific interactions, you're able to target to your specific tissue, liver with N-acetyl galactosamine. But then the CDM masking agent is designed to fall off, releasing into the endosome, which then allows your peptide to interact with the membrane to release the siRNA. And so this is how we believe our DPCs work. Kind of as just an alternative picture schematic. But when they're co-administered, they just end up arriving at their respective cell surface targets with similar kinetics, or even if it's slightly off through endosomal processing, they'll end up in the same endosomes. The DPC, this binding to the cell surface, triggers internalization. And part of the receptor biology is the internalization trigger in the maturation of the endosome results in a low pH compared to plasma. And that triggers the release of our peptide, and that is then, the peptide is then able to promote the endosomal escape of the RNAi trigger into the cell cytoplasm. And then once you got the RNA into the cell cytoplasm, that's when it can engage the RISC machinery so that you can get knockdown of your target gene expression. And so we actually work with a peptide that was originally kind of derived off of melittin. So this is one of the classic cationic amphipathic peptides that's out there. It's a naturally occurring peptide, a major component of bee venom. It's not the component that people tend to be immunogenic against, but it is a major component in there. And so we refer to our peptides as melittin-like properties, as having melittin-like properties because of their similarities in structure and function. So how does melittin work anyway? Melittin structure is one of those things that have been studied a lot. There's over 2,000 references just on structure and function of melittin and its methods of interacting with membranes. And so, but to really simplify it and kind of put my finger on which one I think, because there's obviously a wide variety of possibilities here. Melittin in solution is a random coil. When it comes in contact with a membrane and induces, it's shown here as like a full amphipathic helix. It's probably not fully helical, but parts of it are, and it's a very amphipathic. Melittin's got a very hydrophobic face on one side, ends up sitting parallel to the plane of the membrane. And if your melittin concentration reaches a certain threshold, melittin will insert to be perpendicular to the plane of the membrane, and as part of that, you'll end up with short-lived pore formations through which our siRNA can escape into the cytoplasm from the endosome. And so here's kind of some in vitro data to kind of just give some ideas, just a general idea of what's going on and why we felt comfortable doing this given an IV injection of our molecule. So MLP itself is very potently interacts with membrane. So here's a red blood cell heme release assay. And when you're in the micrograms per mil, it's a couple single digit micrograms per mil to release about 50% of your red blood cell heme. It's at physiological pH in 1 hour time frame. But if you modify it with CDM-NAG, we're then unable to detect any red blood cell heme release after 1 hour of incubation with the red blood cells. So and what's happening here is our CDM moiety, which is a disubstituted anhydride, maleic anhydride, is reacting with the lysine amines on our peptide and the N-terminal amine, and it's neutralizing the cationic charge. And with that cationic charge neutralized, it just is unable to potently interact with the membrane anymore. And then once you've kind of prevented those interactions from occurring, then you have a hydrophilic sugar molecule that can then act as a targeting ligand. And so when we're talking about targeting to the liver, that usually universally means NAG targeting. It's where It's a high affinity ligand for the ASGPR receptor. So ASGPR is a receptor expressed on hepatocyte cells. Its role is to clear serum glycoproteins from the plasma. It does this through clathrin-mediated endocytosis. And as part of the receptor internalization, there's a late endosome pH drop that is important for our molecule, but at that point, the receptor usually releases its ligand, and it's recycled back to the cell surface, whereas whatever it internalized is then processed into a lysosome for degradation. What makes ASGPR such a nice receptor is that it's highly expressed in hepatocytes. You have somewhere between 0.5 to 1 million receptors per cell. And it's also rapidly internalized upon binding, and overall it has a recycling time of about 15 minutes. So this is a kind of example of one of our earlier data points. It's a conjugate in this point, so sorry for that kind of switcheroo. But if you have N-acetyl galactosamine ligand, NAG delivery of DPCs, you do an IV injection, in this case a mouse. You wait an hour and then you section the liver and look for, in this case it was fluorescently labeled siRNA attached to the DPC with the NAG targeting ligand on it. You can see this internalization as endosomes here. If you don't use N-acetyl galactosamine and instead you use a glucose ligand which does not bind to hepatocytes, you end up with very little signal inside the hepatocytes. And so this is just an example to show that the DPCs are compatible with N-acetyl galactosamine targeting, and that it results in increased hepatocyte targeting delivery. Right, so once you're inside the endosome, then there's this pH change. And so here, I'm going to show, like, the CDM is designed to fall off. And so here's a red blood cell heme assay that I just showed at 1 hour. You can have up to 1,000 micrograms per mil of the peptide without seeing any red blood cell heme release. Yet at pH 5, and this is, sorry it's not on here, it's about 20 micrograms. Within 10 minutes you have a half-life of the heme falling off. And it turns out that half-life of the heme release in this in vitro assay is about 10 minutes at pH 5. And there's 5 CDM-NAGs on the MLP. There's about a half-life of each individual CDM-NAG falling off at about 2 minutes. This is also telling us that nearly all the CDM-NAGs need to fall off in order for this to be potent, for it to restore activity with the membrane. All right, so now I'm going to show you some target gene knockdown with our cholesterol-RNA trigger moving in vivo with our NAG-MLP DPC. In this case, we're injecting ICR mice with and what we're following is serum factor 7. So factor 7 is a clotting protein that is synthesized in the liver, but is then excreted into the plasma. So this is a really nice screening program because you can just take a blood aliquot every so often and measure the clotting rate of the protein. And so the target gene knockdown, cholesterol-RNA by itself does not give great knockdown, but you can actually use 10-fold less and add the NAG-MLP, and you end up with 99% knockdown here. So by going 1/10 the dose, you can go from about 20% knockdown to 99% knockdown. But if your RNA doesn't have a cholesterol to make sure it gets to the liver effectively, you have very little knockdown. It's sequence dependent, so if your RNA isn't targeting the gene or the expression level of the protein that you're following, you don't see that knockdown. And then if your endosomal peptide isn't masked with a targeting ligand, but instead something nonspecific membrane, something nonspecific but not targeting like PEG, there's also very little knockdown. But if you use the NAG-MLP and the cholesterol-RNA, you end up with a very potent gene knockdown. And so this is, again, normalized serum factor 7 dose titration of the siRNA. And so whereas there's at 10 mg per kg, 20% knockdown there, at 0.03 mg per kg, you get 80% knockdown, and then at 1 mg per kg, you're ending up with about 99% knockdown. So I figured we like MLP because it's rapidly biodegradable, but I figured this would be a good data point for the crowd, just some comparisons of the biodegradable L versus the D. The L peptide and the D actually turned out to be equally efficacious. So here's where we made a NAG-MLP conjugate with the D peptide, and then here's where we made a NAG-MLP conjugate with the L peptide, both injected with the same cholesterol targeting factor 7. In this case, we're actually looking at the mRNA as opposed to the protein expression. And now they're both actually cleared from the plasma at identical rates, but whereas the DMLP is targeted to the liver and then stays there, the LMLP is able to carry out its function and then is rapidly metabolized and eliminated. And so this is, sorry, I forgot to mention just iodine labeling through Bolton Hunter. And so if you look at 24 hours post-dose, nearly all of the NAG-MLP has been degraded and excreted, whereas the D peptide is still very heavily in the liver. So we have this live imaging. It takes place over about a 4-hour time period that captures kind of the arrival in the liver and then the biodegradation. So it's a positron emission tomography that we carried out on the UW campus through their live imaging facility. So if you could play the video, that'd be great. And so you can see here they're both arriving at the liver in similar amounts and similar time frames, but then after a short period of time, the peptide is degraded and then renally excreted. And so whereas the D peptide ends up sticking in the liver for a much longer time. And so we decided to move forward with the LMLP here because if they're both equally active, you go with the one that's more rapidly cleared. That doesn't seem to be showing up well. So yeah, my apologies. So there are things here. So the Arrowhead Pipeline, I'm going to talk about the ARC-520. It's a little hard to see, but that's for the Hepatitis B program. Thank you. See, I promised you. We have a follow-up drug, ARC-521, that I'll mention, kind of the reasoning for that. We also have an alpha-1 antitrypsin deficiency disease drug, so that's a rare orphan disease of a globulin formation due to a misfolding disease of a protein that's expressed in the liver. We have factor 12, which is we're using for thrombosis and angioedema, also a liver program. We're also looking at LPA. So LPA is a genetic risk factor for cardiovascular disease that can't be impacted through diet or exercise. So, and meta-analyses suggest that it's a really important one for cardiovascular disease. And then outside the liver, we have a program against clear cell renal cell carcinomas where we're aiming to knock down the HIF2 alpha gene in those cancer cells, since that's part of the mechanism by which those cancer cells tend to occur, or part of the reason they become cancerous. So I'm going to talk about, in a little more detail, some hepatitis B virus and our program with it. And so hepatitis B is obviously a pretty nasty disease. And so there's definitely a clear need for better therapeutics. So there's about 360 million people with this chronically infected worldwide. So I'm very specifically using chronically here. There's acute infections as well, but we're mainly talking about chronic infections here. So there's about 500,000 deaths annually due to hepatitis B virus related issues. And two of the major ones are cirrhosis and hepatocellular carcinoma. So it's estimated about half, or you can find people claiming anyway that it's about half of hepatocellular carcinomas are due to HBV. One of the complicating factors for these chronic infections is that the immune system and the virus have a complex interplay, and so what happens is referred to as immune exhaustion. The virus is able to make and output so many proteins that it's unable to continue fighting the virus through your normal T cell mechanisms. And so one of the kind of beliefs in the field, and what kind of goes with small percentage people that are on existing drugs, is that if you can figure out a way to knock down the S antigen, which is one of the viral proteins that's made, and clear that from the person's serum, that's what seroconversion is referring to, then you've reached a functional cure. Functional cure usually requires, there's definitely a long waiting period between after treatment for that as well. And so existing drugs for hepatitis B virus, the kind of standard of care that people have right now, are either reverse transcriptase inhibitors or PEG interferon. Both of these are, have problems. Reverse transcriptase inhibitors can improve the inpatient outcomes but require lifelong treatment, and they just aren't very pleasant to take for your entire life. And they don't significantly impact your ability to become functionally cured. PEG interferon is, on the other hand, has very significant side effects, but it does actually give you an increased chance of seroconverting so that you can stop taking drugs. So the kind of standard cartoon for our hepatitis B virus is that there's this HBV variant, and it's able to infect your cell, and so that leads to the expression of a cccDNA inside the nucleus. That cccDNA is able to generate mRNA. That mRNA leads to more replication of the viral DNA, as well as a large number of viral proteins. These can recombine and then lead to more contagion and reinfection, but these viral proteins themselves are also exported into the bloodstream. And these viral proteins in your bloodstream are what lead to an immune suppression. And so without the and so eventually this leads to liver cancer, cirrhosis, or death in a large percentage of the population. And so with ARC-520, just to go back one. And so, reverse transcriptase inhibitors are able to block this, but not so much the viral protein expression. And so what we're hoping with ARC-520 is because if we're able to reduce the mRNA expression as a whole, then we will no longer have the cccDNA or the viral proteins. And so our goal is to have a short window treatment period that will reduce the viral antigens in the bloodstream, allowing your immune system to recover. And then from there, you'll lead to seroconversion through reactivation of your immune system. In here, we're defining that through S antigen seroconversion. So our screening strategy, when we pick a target gene that we want to go after, in this case, the HBV DNA, is to bring up a schematic. You can look at, this is just the whole thing. You can look at the core here. This is one of the proteins that's expressed. So these are just different proteins that can get expressed by the DNA. And we basically make sure to stick in the targeted conserved regions, so what's genomically conserved across the different genotypes of HBV. And we chop up all those regions into the kind of RNA duplex trigger design screening system, and then do a in vitro cell culture system. And so here's kind of the variability that you'll get based on using the same RNA duplex design, but just hitting different parts of that viral DNA design. And so, what we do is we take the ones that are most popular, most potent here. They became popular because of that, but it was the potency that we chose to bring them into animal models. So humans are really the only ones that are natively infected by HBV, so that makes studying HBV infection a lot more difficult. So we decided to move forward with a non-transgenic mouse model for HBV infection. This is more along the lines of an acute infection as opposed to a chronic, though. So we made a plasmid of the HBV DNA, and we do a hydrodynamic tail vein injection of this plasmid. So if you're not familiar, it's a high volume, rapid tail vein injection that then leads to the HBV genome entering into the liver hepatocytes. And so after that hydrodynamic tail vein injection, about 5 to 20% of liver hepatocytes you'll be able to detect HBV RNA and DNA and antigens generated in the liver. And then also you'll be able to detect in the blood the S antigen, the E antigen, and other viral particles that get secreted by the liver. 3 weeks later, a normal IV, we give it an IV injection of our NAG-MLP along with our lead siHBVs. And then we assay for HBV knockdown through blood collection. So I'm showing here co-injection of our cholesterol-RNA that with the NAG-MLP, and in this plot we're going to follow the S antigen. Here you can see the kind of slow clearance because it's an acute infection of just the mouse over time when it's injected with isotonic glucose. But in contrast to that, we see very deep knockdown and sustained knockdown with both of these two triggers, 74 and 77. This is true for the S antigen, where you see about 3 to 4 log reduction, and you're staying over 2 log reduction for over a month. You have HBE knockdown that went below the limit of detection until we stopped analyzing it, which was just the end of the study. And then the viral HBV DNA, you also see significant lockdown for about 1 month. So because it covers a wider known variety of HBV genotypes, we decided to go not with 1 RNA, but 2. So it's 2 different RNAs that are in that we moved forward with a constant mass ratio with that of NAG-MLP. And so the NAG-MLP in this study is either 3 or 6 mg per kg. Then it's co-injected with cholesterol-74 and then the cholesterol-77. And then each one will be 1.5 plus 1.5. And so if you do that with the S antigen reduction, you get 1.5 logs of knockdown. And then with the 6 mg per kg of the NAG-MLP, with 3 of each of these, you end up with about just short of 3 logs of knockdown. So from there, we actually were lucky to get into at San Antonio a chimpanzee facility, and we're able to do a number of ARC-520 studies on them. And so these are lab-created infection animals. They are chronically infected at this point, though. People have been studying them for a long time, trying to cure them. And so there was 5 males, 4 females. They had 9 to 37 years old. And they actually, what is representative of what you see in humans, there's 2 major serotypes that you can characterize them by. There's where you actually see large amounts of the HBE antigen, and then so the HBE antigen positive, or where there's a lot HBE negative, where there's a lot less of that antigen present. One of those chimpanzees actually converted from an HBE positive to an HBE negative serotype during our nuke lead-in. So this was a ARC-520 plus nucleoside analog. And so we gave daily nukes, and then we had monthly injections of our ARC-520 from 6 to 11 months. And what I think was most important that we saw here was that there was a greater S antigen knockdown in the HBE antigen positive serotype compared to that of the HBE antigen negative serotype. And so the S antigen knockdown was about 2.1 logs for the E antigen positive chimps, and in the S antigen knockdown in HBE negative chimps was only about 0.7 average. So we've been doing a lot of deep sequencing through liver biopsies of these chimpanzees, and what we found is that in the S antigen, in the E antigen positive chimps, nearly all of the DNA that you find in the liver is actually in the CCC DNA form, whereas in the E antigen negative chimps, nearly all of the liver DNA is now actually into an integrated DNA, is integrated into the host DNA, but it's only a fragment of the DNA that gets integrated. So part of the consequence of that integration is that our ARC-520 RNAs are no longer targeting, they don't target the integrated DNA. But the other thing that was really interesting that we saw is that 7 of 9 of the chimps exhibited indications of immune reactivation. So this wasn't related with the timing of the treatment dose, which is also a concern. But what we were able to see is that as the serum HBV DNAs were lowered, we could see induction of the CXCL9 and CXCL10. And so one example that we had, it's our best example, was chimp Minetta, so that's the name of the chimp, had sustained CXCL9 activation. And this was actually associated with a therapeutic ALT flare, so that's a sign of liver immune activation, like your body attacking your liver cells, which is what we want to happen. And so after we halted treatment on Minetta, we saw a temporary increase in HBV serum DNA, but that was then followed by an increase in the CXCL9 and CXCL10. And then so following that, there was then a subsequent decrease in the serum DNA. So without our doing anything, you could see the DNA start to rise up, go back down. And it was concurrent with the CXCL9 and CXCL10, suggesting that there's some T cell activity there. And so 31 weeks after we stopped treating him, he still showed sustained virologic response. So we're hoping this is the first of many examples where we have seroconversion or something looking like it. But the evidence of the immune reactivation, which is crucial to our understanding of how to cure people in HBV, is pretty exciting there. And so with ARC-520 in chronic HBV patients, we've looked at E positive, we've looked at E negative patients, and what we saw in the chimpanzees actually correlates to that of the humans. And so if you follow the knockdown of the HBe antigen positive patients, which from our chimpanzee data we'd predict to be all cccDNA or heavy cccDNA, you can actually see multi-log knockdown of the S antigen. Whereas with HBe antigen negative, which we'd expect to have high levels of integrated DNA, we saw very little knockdown. And then there was one seroconverter in our human trials that was intermediate. So this was all, and this is our phase 2 data, so we had to get through all of the phase 1 data to do all this, but I tried to stay a little high level here. So as in chimps, the HBe antigen negative patients likely produce S antigen for the integrated DNA, but is not targeted by the ARC-520. But in order for people to be cured, we actually really need to knock down that S antigen. And so that's where so what we've learned anyway so far is that ARC-520 is showing good single dose tolerability. We've injected 84 humans so far, single dose, and now we have multi-dose tolerability in 9 chimpanzees. We've still had no serious or severe adverse events or dropouts due to adverse events, and then no suspected signs of end organ toxicity through our kind of clinical chemistry and things like that. What's really exciting is that we can see that ARC-520 provides deep and sustained knockdown of the cccDNA-derived mRNA and proteins, and so that includes a lot of proteins that the E antigens, core antigen, and S antigen that are all really important for patients. So we were really excited to see this because it's really telling us that the ARC-520 DPC is actually doing what we designed it to do. And so moving forward, we just need to figure out what's the keys to getting that immune reactivation in humans. So what's the dosing strategy? How deep and how long do you need to have knockdown for that to occur? And so just as a quick note, we don't, this is where our ARC-520 follows in. It's now specifically designed against the integrated DNA. And we just announced that we've injected the first patients in a phase 2 trial of ARC-521. Right. So this is obviously a ton of work. So it's really exciting working at Arrowhead. I'm really happy to be there, and I'd really like to thank again being invited, and then all of you guys for listening. And please, if you have any questions, happy to take them.